Yulia, you can now grab the screen. And Krasi and Yulia, now the floor is yours.
Thank you, Daniele. So shall I start?
I think we have 2 minutes to go. It's 28.
Usually people tend to join like exactly on the minute. Maybe wait for 1 minute and then.
Yeah, it's still 28.
I think I should start.
Please go ahead.
Yeah. So welcome everyone to the second session, the second parallel session of the first day of this digital edition for the International Conference of Mathematical Neuroscience. The topic of this session is cellular and subcellular dynamics. And our first speaker today is Yulia Timofeeva from the University of Warwick. And Yulia will talk about computational modeling framework to study calcium activation of synaptic vesicle fusion. So Yulia.
Thank you. Thank you, Krasi. And thank you very much to the organizers for inviting me to give this talk. It's clearly new experience in this current time. And I really enjoyed the first part of today. So I'm going to focus on the single cell. And I'm not just going to focus on the single cell, but I'm going to focus on the presynaptic bouton. And synaptic transmission will be my main focus. In many parts of central nervous system, these presynaptic boutons are quite small. So we're talking about 1 micron in diameter. And this imposes limitation on experimental methods. The for realistic computational modeling of presynaptic terminals in general and the release machinery are essential tools that allow us to bypass the limitation of experimental approaches. And as you can see later, I'm not actually going to focus on the whole presynaptic terminal. I will be only focusing on the fusion machinery. So this is a brief overview of my presentation. So first of all, I will discuss biological background and highlight some motivation for this work. Also, I will introduce some phenomenological models in this field, and then I will introduce this novel mechanistic, so-called dual clamp model of synaptic vesicle fusion that we developed. So this work has been done in collaboration with Chris Norman. He's a PhD student at Warwick, and also with synaptic neurophysiologist Kirill Valinsky from UCL, and biochemists Shyam and Jim Rothman from Yale. So first of all, I will remind quickly the main processes behind the synaptic fusion. So when the action potential invades the presynaptic terminal, we have the activation of voltage-gated calcium channels, and the calcium, we observe the calcium influx into the terminal. Because of this presynaptic elevation of calcium, this calcium elevation will trigger vesicular exocytosis, and the neurotransmitter will be released in the synaptic cleft. And clearly we know that after that, this neurotransmitter quickly diffuses to the postsynaptic site, and the signal will be transmitted from presynaptic cell to the postsynaptic cell. So what I'm going to focus here actually on this part of the vesicular exocytosis. So we can see here that during this process, we will observe a number of synaptic vesicles filled with the neurotransmitters. These synaptic vesicles will be docked at the so-called active zone in the presynaptic terminal before this process of exocytosis will take place. So again, to shrink it down to the scale of the system I'm focusing on here, I'm focusing on the fusion machinery. So we have a synaptic vesicle here, and this fusion machinery, which will allow this particular vesicle filled with neurotransmitters to release these neurotransmitters in the synaptic cleft. So this will be the scale of the system I'm looking at. So what can we say here in terms of the synaptic fusion in general? So it is well established that synaptic vesicles fusion with plasma membrane is mediated by so-called SNARE proteins. And SNAREs can constitutively assemble into a stable complex when during this assembling state, the SNAREs bring opposite membranes together and provide the energy required for fusion. So what we will observe here, that if we look at the presynaptic dynamics and if we look at the presynaptic terminal, neurotransmitter release will be a very fast process. So this release will be at the scale of 1 millisecond. However, similar processes, when we are talking about constitutive vesicular fusion, this process is much slower at the scale of 1 second. So one big question is, how can the fusion machinery operate 1,000 times faster at synapses? So before we go to the modeling part, let me provide more detail about what is known about the fusion machinery at the synaptic level. We have a synaptotagmin, which is a calcium release sensor. This synaptotagmin plays an important role in neurotransmitter release and this fusion process. So it is believed that at the rest, this synaptotagmin will clamp SNARE complexes. During the action potential, when we have a calcium elevation in the presynaptic terminal, this calcium binding can release this clamp and the fusion will take place. This is the general idea behind this process. However, this happens, how this happens still remains a mystery. What else do we know about the synaptotagmin in this process? So when calcium enters the presynaptic terminal and bind to the synaptotagmin, this will trigger membrane insertion of synaptotagmin in the cell membrane, and this believed to reverse the fusion clamp. There are two binding sites at the synaptotagmin, and it's believed the C2B domain is essential in this process. Also, if we look at the recent data from the crystal structure, we can see that up to 2 synaptotagmin molecules can bind to a single SNARE pin. And what is believed that there is a primary site which will bind synaptotagmin 1 at the SNARE pin, and there is also the tripartite site which can bind either synaptotagmin 1 or other isoform of synaptotagmin. For example, a key player here is synaptotagmin 7. And the main of this work is to develop a realistic model that will allow us to study calcium-driven vesicular release during physiological patterns of activity. So what we would like to use this model for, we would like to, to use this model to discriminate between different molecular mechanisms, because there are still many uncertainties how this process occurs. And also we would like to have this model with the predictive power. So we would like to have a mechanistic model so that we can actually study different mutations in the fusion machinery. And these mutations are known to be linked with the neurological disorders. So first of all, let me introduce what is known in terms of the phenomenological models. There are a few phenomenological models that have been widely used in the field. One of this model is the so-called 5-site model. So here you can see that in this model, up to 5 calcium ion can bind to the sensor. Again, I would like to stress here that this model is phenomenological. So the vesicle simply has a sensor which can, and calcium ions can bind to this sensor. So, and the release can occur only when all 5 calcium ions will bind to this sensor. And typically, this type of models are developed not for the small synapses, because it's very difficult to measure anything in the small central synapses, but these models are developed for the calyx of health, which are much more bigger. And then the data collected from the calyx of health, typically it will be peak release rate as a function of peak calcium, and then the model simply is fitted to the experimental data. So after this model has been introduced, another model that has been widely used is the so-called allosteric model. It's kind of a generalized version of that 5-state model. So here we can see in the kinetic scheme that the fusion can happen from many states of the sensor, but clearly the fusion will happen with the higher release probability if all 5 calcium ions will bind to the sensor. And if we look at the 5-site model, clearly the 5-site model that I described in the previous slide cannot explain the release rate for the low calcium concentration. This allosteric model can describe the experimental data even for the low calcium concentration. So this model has been widely used, and I also used this model previously in some of my other studies. Also later, this so-called dual sensor model was introduced. You can see this kinetic scheme is much more advanced. And here we can see that there will be different states for the so-called synchronous and asynchronous release. I didn't mention it, but in case somebody might not know, synchronous release, it's a release straight after the action potential, typically in the window of 10 milliseconds. And however, the fusion can occur even in a longer time scale, and this release at the later time will be assigned as the asynchronous release. So here you can see in the dual sensor model, we can discriminate between synchronous and asynchronous release and explain the experimental data. Again, the data we used from the calyx of health, the large synapse. So here we would like to introduce a mechanistic model. We would like to introduce the synaptotagmin and the fact that when calcium ion bind to the synaptotagmin, it can insert into the membrane. So the initial scheme at the level of synaptotagmin is shown here. So synaptotagmin can bind up to 2 calcium ions, and when this takes place, this synaptotagmin can enter into the membrane with some rates K in. So this is the general scheme, and we can see we have a number of parameters here. And because we are looking for a predictive model, experimentally constrained model, clearly, first of all, we need to constrain these parameters. And again, it's not so easy because at the level of the synapse, not all parameters can be estimated reliably. So if we consider only K-in and K-on, and K-off rates, what is known that in solution, both synaptotagmin isoforms, so synaptotagmin 1 and 7, show similar calcium binding affinity. So this is in the absence of the lipid membrane. So we're looking at the kinetic scheme at this level. And also it's known that calcium binding to synaptotagmin is very fast and limited by diffusion. So if we take this fact into account and consider this KD parameter for both synaptotagmin 1 and synaptotagmin 7, we can then constrain the K-off variable. Now we have to look at the K-in and K-out rates. So again, if we look at the experimental data, what we know that both synaptotagmins will show different membrane dissociation kinetics. And when the experiments are conducted to measure this parameter, what it's done that it's really turned to be measuring some observed rate, as showing here as K observed, which make a transition from this membrane inserted state to the state S1 when we have only one calcium ion bind to the synaptotagmin. Also, we know that the process of loop insertion into the membrane, it's a fast process. So if we take this into account for the K-in parameter, and if we take this experimental data on the observed rate, K observed, we can actually constrain our K-out variable here. And as we can see here, the dissociation kinetics will be different. So we will observe different rates K-out for both synaptotagmin 1 and synaptotagmin 7. So one more thing that we need to consider before we construct this whole ingredient for the single vesicle is the release rate. So fusion of synaptic vesicle with the presynaptic membrane is resisted by repulsive forces with the energy barrier E0. This energy barrier again was estimated experimentally. Also, it has been estimated that each assembled snare complex provides delta E energy to overcome this barrier. So again, from the experimental estimates, we can know the value for this delta E. So here it's proposed to consider the rate at which N-free snare pins overcome the barrier, as described here by this Arrhenius equation. And we have additional parameter here, A. However, using the fact, again, from the experimental studies that the single snare pin can mediate fusion in vitro on a time scale of about 1 second, we can constrain this parameter A. So we can see here that we will have a function for the release rate, which will be a function of N free snare pins. So now, if we look at the level of the single vesicle, again, we know the following, that structural model suggests that it might be this magic number of 6 that is required to support the synaptic vesicle in this clump state. And later, new experimental data came using this cryo-AT technology, which again demonstrate that it's probably this magic number 6 snare pins. They're holding this synaptic vesicle in a dock state and will lead to the release of neurotransmitters. So we took this on board, and now we'll put together all this mechanistic model. So we consider snare pin, and we consider there are 2 sites, primary sites and tripartite site. As I mentioned earlier, primary site can be occupied only by synaptotagmin 1, but the tripartite site can be occupied by synaptotagmin 1 or synaptotagmin 7. We have a release rate, which will be a function of unclamped snare pins. And also we assume that snare pins can fully zip and then drive the fusion only when both clamps are released. So the clamps from the primary site and from the tripartite site. So this is the general mechanistic framework for our model. So now we would like to compare it against the experimental data. As I mentioned, it's difficult to get any data in the small central synapses. We have only the data from the calyx of Held, and the allosteric model is a good model to describe this data. So our benchmark was just this allosteric model that was introduced earlier for comparison our results with this model. So here what we can see, we considered 3 limiting cases. And the first scenario we were interested in, which is typical for the experimental setup, is to consider different calcium transients in the form of the calcium step function. So here I'm showing some results for the step function for the particular calcium value. And the first limiting case is when we consider that only primary site is occupied, and this will be occupied with synaptotagmin 1. So here we can see that the dashed curves will be showing the results of the allosteric model from the calyx of Held. Again, 2 different curves describing the data in the calyx of Held from 2 different experimental laboratories. And there our mechanistic model with a single primary site demonstrate these blue results. Now, if we consider the case when both sites are occupied by synaptotagmin 1, so-called SIT1 scenario, so we will observe this decrease in the release rate as a function of time in our mechanistic model. However, we consider the scenario when we have SIT1, SIT7 combination, so the tripartite site is occupied with synaptotagmin 7, so we observe the increase in the release rate. So if we summarize it in a single plot, typically for the experimental data, peak release rate is the concentration of calcium. So we observe the following. So in the first scenario, synaptotagmin 1 can describe sufficiently well the kinetic and calcium cooperativity. This is the blue curve. If we introduce SIT1 scenario, we actually observe a much steeper curve than the experimental data. But introducing back synaptotagmin 7 at the tripartite site will explain the experimental data very well in the calyx of Held. Also, what is interesting, what we wanted to find out, what is the predicted number of snare pins that will be required for the fusion. And here again, we did some computation to check it. And what our model predicts is that 3 snare pins are driving vesicle fusion and physiological calcium concentration. And this is again a very good agreement with the experimental data, which is suggesting that to provide sufficient energy to maintain the millisecond rate of the synchronous release, 3 or 4 release snare pins are required. Also, we could observe this magic number 3 only in the presence of synaptotagmin 7 in the model. Again, this will indicate that synaptotagmin 7 plays a key role in this fusion dynamic. So far, I discussed the experimental data when we consider calcium transient as the calcium step function. Now I would like to briefly mention the process of short-term facilitation, because the idea of this model is to have some predictive power, and we would like to use it describing some mutation in future. So in terms of the short-term facilitation, typical protocol when the 2 action potential are applied, and then the experimentalists are observing an increase in the release rate and the second action potential. Also, experimental data indicate that, say, if synaptotagmin is absent in the system, so short-term this can suppress the short-term facilitation, again indicating the synaptotagmin 7 is important. And also my collaborators, Kirill and Sham, introduced this molecular fusion model of cooperative activation, which suggests that the combination of SIT1 and SIT7 might be important of explaining this facilitation of the synchronous release, because both synaptotagmin 1 and synaptotagmin 7 will be operating at a different time scale. So the proposal of this molecular fusion model is that SIT1 combination will be important for the synchronous release, and the combination of SIT1 and SIT7 will be important for the asynchronous release, as well as for the facilitation at the second action potential. So taking this into account, we wanted to test our model when we apply 2 action potentials and we introduce the realistic calcium transient. So what we can see here in this framework, and because calcium transient will not be known from the experimental data, but again, in the previous work, we developed realistic 3-dimensional models of calcium dynamics in the presynaptic terminal. So we simply took this calcium transient in the active zone near the synaptic vesicle, and we use this in our framework. So at the level of the first action potential, again, we have some calcium transient, and we can compute the release rate and the probability of release. And we can do the same at the second action potential. So now if we summarize these results, paired pulse ratio as a function of peak calcium concentration, we will observe what we wanted to see. So if we consider the situation SIT1, SIT7 in our framework, we can reproduce experimentally observed short-term facilitation. But we will not observe this facilitation for the SIT1 only, in a limiting case, or SIT1 situation. So here, I would just like to summarize the main idea behind this modeling framework. So we introduced this mechanistic model, dual clump model, based on the known results from the experimental studies. What our model demonstrated that SIT1 binding to the primary site is sufficient for explaining the kinetic and calcium cooperativity of synaptic vesicle fusion. We observed that if we have SIT1 situation at both primary and tripartite site, we start to see some divergence from the experimental data. However, if we consider SIT1, SIT7 combination, this framework will explain the experimental data very well in the calyx of health. And also the short-term facilitation can be explained well with this SIT1, SIT7 combination in our mechanistic modeling framework. So in future, we're planning to use this model to study more different mutation at the level of the synaptic machinery. And also we consider other possible scenarios at the level of the molecular dynamics here. So I would like to stop here, and thank you very much for your attention.
Thank you very much, Julia, for very interesting and clear talk. We seem to have a question from Victor Matveev. Not surprised. I was expecting that he's going to ask the first question. Julia, great talk. Could you please clarify the very last step in your full mechanistic model? How is the last irreversible fusion step implemented exactly in terms of the ODE or reaction description, given the 6 snare pin complexes?
No, this is all simulated stochastically, not in a deterministic framework. So it's all stochastic simulation. We use a Gillespie model, Gillespie algorithm to simulate the model, and we simply probabilistic transition in the Markov chain type model. In this framework, we didn't consider the special cooperativity between the snare pins, but we are planning to do it in future.
Okay, there is another question. Can you also produce depression, not only facilitation with your model?
So far, my collaborators mostly were working on the excitatory synapses, and we're focusing on the facilitation. Possibly, we haven't done it yet, but because we have this framework now, so we're planning to use it in more studies.
Yes, thanks. I have one question. Why do you think there are 6 snares when only 3 or 4 are sufficient for the secretion to happen?
Oh, it's difficult to say. Difficult to say from the biological evolution point of view. So it might be some kind of backup scenario. Plus, I mentioned this 2 population of snare pins, SIT1 and SIT1, SIT7. So in some sense, there will be different number of pins in these different 2 populations, plus it might vary from synapse to synapse. This is something we would like to explore more in future, but I wouldn't able to add more about this magic number of 6 at this point.
Thank you very much. Thanks again, Julia. I think it's now a good time to get our next speaker, Kyle, to share his presentation.
Okay. Fingers crossed that this works. Share. Oh, let me do this first.
Yeah, we can see your screen, Kyle.
Great.
But not your presentation.
Okay, hang on one sec. Can you see it now?
Yes, that's great.
That's it.
So I'd like to introduce our second speaker for today's session, Kyle Wedgwood from the University of Exeter. Kyle will be talking about multi-spike rhythms in a delay-coupled neuron.
Great stuff. Thank you very much. I'd like to begin by thanking the session organizers and indeed the whole conference for putting this together. It's been really fascinating so far, and I'm sure it will continue to be for the rest of today and tomorrow. Also, just in case I forget, I'm sure I won't, but in case I do, I'd like to thank the people I worked with on this project and the funders for giving me the opportunity to actually do some experimental work. Okay, so I'm going to talk today about multi-spike rhythms in just a single cell to look at sort of what are the capabilities of encoding memories in a very simple system. So I'm going to present our modeling results and then go on to talk in the second half of my talk about experimental results. But I just want to say that these are quite preliminary because they happened about 4 days before the lockdown was announced in the UK. The labs are starting to open up again, so I'm hoping to collect some more results. But I really just wanted to give everyone a flavor of what we're doing and to seek any feedback that anybody might have. So I'm going to start by talking about a laser system, which may seem like a slightly strange way to begin a talk about neuroscience. But the reason I'm doing this is because it echoes a lot of the ideas that I'm going to talk about. And these kinds of things have been studied in a lot more detail in a laser system. Okay, so it doesn't really quite require an in-depth knowledge of lasers. I'll talk you very briefly through what's going on here. So this is a driven laser system. So the master laser here is driving a slave laser here, such that the slave is phase locked to the master laser. And there's a pulse modulator sitting in between here, so that this can provide perturbations to this phase locking. Okay, so downstream, we measure the activity of the slave laser. So that's what's shown on this screen here. And importantly, the output from this slave laser is split by a beam splitter here, goes down here and is reflected back to that same laser. So this actually is now a recurrent feedback loop, and it has a delay that basically is the time it takes for the light to go from here to the mirror and back again. So what happens, so here's a slightly simplified schematic of just the bit that's important. So this is really just isolating the slave laser system. So you can imagine that there's some external perturbation that I can put in from here, but this is really the feedback loop that's important. Okay, so this represents here the delay, and in this system, the delay here can be tuned because the light actually passes through a fiber optic cable. So if you make the fiber optic cable longer, you make the delay longer. And so what happens then if you give it some external perturbations is you can elicit pulses from the slave laser. And so here the delay, so I'm just going to minimize everyone on my screen. So the delay here is tau. So that's the time it takes for the light to go from the mirror, get reflected back to the laser. And if you elicit pulses in this slave laser, then you can essentially, these can be repeated over time, because the light that's coming back to the laser at some delay tau then exhibits this recurrent excitation. So here we can see on the y-axis this round trip number. So essentially you just take a long time series and chunk it up into epochs of length tau, which is the delay, and just essentially stack them on top of each other. So this is in the laser field, they think of this as a pseudo-spatial variable. And so what you can see here is that after sort of maybe 105 maybe round trips, we see a stable pattern of 6 pulses per delay period that basically just goes on forever. So this is a stable, robust pattern. And the idea is that in the laser community, people think of these as memories, so memories that can be stored by the laser system. And in actual fact, as the pulses are triggered, we can store a variety of patterns, and they coexist, and these are all stable. So we can have patterns with 1, 2, 3, 4, 5, or even 6 pulses per delay period. And the only real thing that we need to have here is that there's some excitation and there's some delay, and the delay needs to be sufficiently long so that we kind of escape this refractory time. So really the question here is, what are the patterns that coexist? Where are they in parameter space? And really we're asking about the capability of this system to store memories. Okay, so that's in a laser system. Of course, we're more interested in neurons. So let's think about neurons. And of course, this is a slightly more traditional thing to think about memories in. So really, what is the capacity of a neuron, a single cell, to store memories? So this is the analog of the laser system, but in a cell. So here we can see the cell body. And of course, as we know, neurons are actually spatially distributed. So here is my axon here. So activity at the cell body will be transmitted down the axon, as I've shown here with these arrows. So if I elicit some spikes at the cell body, I will see them a little bit later at the axon. And I'm just assuming here that the shape of the spikes is the same at the axonal bouton. So there's no attenuation of the signal. But of course, the fact that this is spatially distributed induces some delay, which again, I'm going to call tau. So the tau here is now the time it takes for a signal at the soma to reach this axonal bouton. So this is one cell, and of course, in a neural network, these guys here would be coupled to the dendrites of some receiving cell. But what we can do in this example to emulate the laser experiment is to couple the cell to itself. So now I've just basically bent the axon back round, and now I can see that there are these sites where the cell projects to itself. And now if we give a signal at the cell soma, it will travel down the axon, again with this time period tau, and then there'll be some synaptic activity here. And if this is sufficiently strong to induce a postsynaptic response, then we can hope to get this recurrent activity in this same cell. So this is what I'm going to call the ORTAPS model. And here is a really simple example of a memory, or pseudo memory, if you like. So in this experiment, this is just simulated data. So I give some applied current at some time, I elicit one spike, and then I don't give any more current. And now what happens is the postsynaptic activity is here induced in that same cell, and activity builds up, and after maybe 3 spikes, I'm back on the same rhythm that I started with. So here is an example of a successful encoding of a 1-period rhythm. So these are the kinds of things that we want to try and understand. So I'm going to begin now by talking about the modeling framework which we use to do this. So this hopefully will be familiar to people who study computational models of neurons. So this is the classic Morris-Lacar neuron. We chose this model because it exhibits quite a lot of the kinds of behaviors that we want to see, and in particular, it's tunable to different kinds of sort of bifurcation scenarios. And it's really nice and simple because it's 2D, and because it's 2D, we can analyze it in quite some detail. So here's the standard Morris-Lacar model. So we have some voltage, which is the thing that we typically plot when we talk about action potentials, and some recovery variable W. And for those that are interested, here are some steady state functions and time constant that go along with this model. This model, of course, is not coupled to itself. So we introduce this ORTAPS model. So S is some ORTAPS induced current. And I assume it has a very, very similar form to W. So there's some S infinity, there's some asymptotic steady state. There's some time constant S, which in this model, we're going to assume that the synaptic transmission is instantaneous. So there is a delay term, which is the delay between when the signal is felt at the axonal bouton versus when it is elicited in the soma. But the actual transmission of the activity across the synaptic cleft is instantaneous. Okay, so we can replace S with just this asymptotic value here. So here's the model, and what we want to know is about, well, what are the memories that we can store in this model? So the classic thing to do is to do some bifurcation analysis, which we can handily do in DD-BIFTOOL. So here is an example of when there is no coupling. So kappa here is my coupling strength, and tau is my delay. So if kappa is 0, there is no coupling. So this is the classic bifurcation diagram for the Morris-Lacarte model. And we can see this S shape here. So this is an equilibrium, and we can see a hot point over here on the right. So this is a bifurcation diagram that will be familiar to people that have studied the Morris-Lacarte model under current injection. As I introduce the coupling, the self-feedback, so as I increase kappa, we see hot bifurcations begin to accumulate. Okay, so they start off being quite close to this upper hot branch, and there's one over here. And as I continue to increase it, all of these guys shift over to the left. Okay, so by time kappa equals 30, there's lots of hot bifurcations on this curve. And this can be summarized here. I've just taken all of these 1D slices up to some notional value of kappa and just stack them. Okay, so you can really see that these hot bifurcations, the rightmost one is shifting to the left, but also they're getting more and more spread out along this axis. All of these hot bifurcations here are of the subcritical type. However, they do elicit periodic orbits that stabilize via fold of limit cycles that I'm going to talk about in just a minute. In a similar way, we can do a continuation for a fixed value of kappa, but changing tau, which emulates what was done in the laser experiment. And again, we see something very, very similar. So remember here, kappa is chosen to be 60. So even at 0 delay, the cell is coupled to itself just with no delay. But we still see a very simplistic bifurcation scenario. However, as we increase delay, then we start seeing this collection of hops. And this sort of mirrors what I said earlier, that the delay needs to be long enough for us to get this memory kind of phenomenon. So we want to be in a situation where the cell is excitable, not oscillatory. The results that I'm going to talk about don't depend heavily on that. We can still compute similar things if the cell is in an oscillatory state. But for reference, we set the cell to be here. So this red plane is where we choose our current density. So the cell is excitable. And that means if we elicit pulses in the undelayed system, we would, the non-coupled system, we would elicit one spike and then come back to rest. So here is an example of what happens then if we fix the value of kappa, and we now, rather than changing the current injection, we change the delay. So each of these curves that we're seeing here, these are branches of solution that are elicited from a hop. This gray point here is the hop, and we can see that we do see lots of stable branches. The green here is the thick green is a stable branch. We see lots of stable branches that all correspond to different firing patterns elicited by this same cell. Moreover, we can see that actually very little information is conveyed in the amplitude of this spiking behavior, which is precisely what we expect in neurons. Typically, the firing of a cell is an all or nothing kind of event, but a lot of information is encoded in the period. So we can see here that each of these different patterns has periods that are very well separated from other patterns. Moreover, if we then look at what happens in the two-parameter bifurcation diagram, so now we're changing coupling and the delay, we can trace out folds of limit cycle bifurcations that basically take us to regions where we can express different numbers of stable patterns. So the number of patterns that we can store is indicated here in these little patches. So here we have 1, then 2, then 3, then 4. And here, in these panels here, we're just taking a slice at kappa equals 60. If we actually look at these solutions, so if we look now, we fix the delay to be 400, and we just look at solutions D and F here, we can indeed see this. So here are two solutions, D and F. I've plotted here, this is twice the delay period, just because it looks a bit nicer. So here we can see an example with 2 spikes per 2 delay periods, so 1 spike per delay period. And here we can see 2 spikes per delay period. And the point is that these solutions exist, these coexist, and along with a whole bunch of other solutions that you can see on these other branches here. The other thing that I want to say is that if you look at this solution here, so these are the Floquet multipliers of this solution. And you can see here there's one that's really, really close to the unit circle. And in fact, we see this for a lot of different unstable branches down here as well. And what that means is that even though these are the asymptotically stable solutions, there are lots of other solutions that are asymptotically unstable, but that we can stay close to for a really, really long time. So if we do an in silico simulation where we now give 2 pulses rather than just 1 pulse, in some scenarios, so the 2 spikes elicited by these 2 initial pulses, but then what happens is that the second pulse then falls away. So actually the solution that we get in the simulation is just 1 spike per delay period. Conversely, if we slightly increase the distance between these two pulses, we can the second spike can persist, and we can get solutions that look like this. And these will persist for a really, really long time. You can see here that this is number of delay periods, and you can see this is what, 10 to the 6 almost. Ultimately, of course, it will give way to the stable solution, but it takes a really, really long time. So these have all of the hallmarks that we might think of as memory. So the ability to store different kinds of patterns, but that decay over some quite long time scale. So this is basically a blueprint for understanding when this ORTAPS model can store memories. So a sort of sensible question to ask, well, does any of this actually happen in a real cell? So now let's go to some experimental results. And I guess some of you may well be familiar with the experimental technique, but as a refresher to those people and as a very, very brief primer to other people, I just thought I'd spend a few minutes just talking about how the experiments actually work. So this is due to an experimental technique called patch clamp. And the idea is that what you do is you take an electrode and you put it in a little pipette like this. So you have one electrode that sits outside of the cell, and this is sitting in some saline solution that tries as best as possible to emulate what the ionic composition in your brain is, basically. So we set up some solution that matches cerebrospinal fluid. The other electrode sits in this solution here, which is now, this composition is matched to be what is inside the cell. And so you land it on the cell, a bit like landing on the moon, and then you sort of suck quite hard on a little tube and you rupture the cell membrane. So now you have sort of a contiguous solution here between the stuff that's inside the pipette and the stuff that's inside the cell. And more importantly, the electrode is now inside the cell. So we've got one electrode inside the cell, one electrode outside the cell. We can measure the potential difference across the cell membrane. And then we can track these voltage fluctuations that we know as action potentials. This is not what I actually did in these experiments, because that's actually a bit rough on the cells. They don't really like it if you blow big holes in them. So instead, I used a method called perforated patch clamp, where we actually put an antibiotic in the cell. And what this does is it just lies as really small holes. So we get a much stabler long-term recording, which is useful if you're trying to track things like memory. Here's an example. So here's my pipette with my electrode sitting inside it. So I landed it on this cell here, and you can record activity that looks like this. So these are, spikes that we know and love. So there's the voltage of this cell over a 5-second window. Okay, so that's half the job done. But of course, these cells are sort of isolated. Okay, and actually the probability of finding a cell that is coupled to itself is pretty small. So actually we rely on a technique called dynamic clamp to get over this. So what dynamic clamp does is it takes that signal that I'm recording from the cell, it amplifies it, and then it runs that output from the cell through a mathematical model. So this is just done on a computer, and the computer here essentially allows the direct interfacing of any mathematical model you want to give and a cell. So the model on the computer essentially computes what current you should inject to the cell to encapsulate whatever description you put in here. And then it basically takes that current and injects it back to the amplifier, and then the amplifier sends it to the cell. So this has been around for a few years now, and it's typically used to either virtually insert or remove specific ion channels. If you get your model bang on, you can also use it to modify specific ion channel properties, such as timescales. So it's really, really nice because it's quite a clean experiment if you really believe in your model. So sometimes the pharmacological experiments or the gene editing experiments can give you effects that you weren't really expecting, but this really will just do whatever the model sets. So in our case, we're going to use it to couple the cell back to itself. So we're going to use it to essentially make this green loop here. So let's go back to the model and think about what it looks like. So here is the Morris-Lacar with the ORTAPS. So now if I color in the different parts of the model, so the blue bits is basically the model of the cell. So this is what the Morris-Lacar model did, and of course any kind of Hodgkin-Huxley model will look like this. So I'm just going to leave this to the cell. I'm not going to impose any other dynamics other than that with regards to the intrinsic properties. And this red term here is the ORTAPS model, and this is what's going to be handled by the dynamic clamp. And just to show you how sort of the workflow works, just to repeat what I said earlier. So we measure some activity from the cell. Because now we're doing some delayed system, we have to store the history in a buffer. It's actually done on a little chip here, which I'll show you in a little bit more detail on the next slide. There's not that much memory on this chip, so I actually use a little circular buffer here to store the values of the voltage in the past. Then this guy here computes this autoptic current, computes the current, and then rescales it, and then puts it back into the cell. And this loop just essentially repeats. It just goes on and on and on and on. And that essentially will do the integration of this term in this cell for me. So here is what it kind of looks like. I guess one of the things that I find really interesting about this is, this is just built on a breadboard. I haven't even soldered anything in. So there's a paper that came out in 2017 that really shows how you could make dynamic clamps really, really cheaply. Before then, they were, you needed a whole different computer, a whole different setup for recording. But this is a really cheap, really efficient way to achieve the same kind of result. It's really fast. So this little chip here is it's called a Teensy 3. Well, this is a Teensy chip. This is a number variant 3.6. But it's really, really fast. It samples pretty robustly at 100 kilohertz. It doesn't have that much memory on it, but it has enough to do what I want to do. And importantly, it's got a lot of analog inputs and outputs. So you can use this to control quite a lot of properties in the cell, or even a couple other cells, which I didn't do, but would like to do in the future. So this is really nice. It runs in Arduino, so it's basically a little bit like programming in C or C++. I wanted to exhibit a bit tighter control over the cell when the experiments were actually going. So I wrote a little MATLAB controller for it, which looks like this. So if anyone's interested in using that little teensy thing, you can download this controller from my GitHub here. And I really like it because I use MATLAB to also do my electrophysiology recordings. So in support of open source software, you guys can check out the Symphony here, which is a great MATLAB package for electrophysiology recordings if you see fit. Okay, so that's all I wanted to say about the setup. So let's go to the results. And I stress again, these are reasonably preliminary, but I think they illustrate the point quite well. So here is an example of what happens if I just patch the cell. I'm injecting no current now, so this is just spontaneous activity, and we can see pretty nice, robust tonic spiking activity. This is not quite what we want, because if you remember in the model, we set ourselves to be on the other side of the bifurcation, so our cells are excitable, so we can just inject a little bit of hyperpolarizing current to essentially put us on the other side of the bifurcation. So this looks like this. So now everything is set up for us to actually run this experiment. So here is the actual experiment. So now you can see the red here is the applied current that is sort of standard electrophysiology experiments. So you just give some square wave pulse or something else. But then I switch it off. So I give it a little pulse to begin with, and then I switch it off for the rest of the time. And in gray here, we can see that the current that is being injected by this little chip here. So this is the dynamic clamp. This is the autoptic model. And what we can see is that even though I don't apply any more current, that the spike shape builds up, and it's actually pretty robust. So this actually happens after just one spike, and then this persists essentially indefinitely. So this is the example of this simple encoding, this simple one spike to one delay period that I talked about right at the beginning. So that's the one spike experiment. What about the two spike experiment? Okay, so this was the next thing that we tried. So this is kind of the same kind of picture we saw before. So round trip number on the y-axis, and then I just renormalized everything so that my delay period is 1 here. So if we try and give two spikes that I'm going to color code with a little orange arrow and a little green arrow here, if we get the delay period wrong, or the coupling is not big enough, then we cannot elicit recurrent spiking activity. So here we can see the two spikes that they're elicited by these two initial pulses, but we don't see any activity after that. Okay, so if I retune it a little bit, so now my coupling strength is high enough, but my interpulse interval is a bit misspecified. So you can see here that the spike that's elicited by the orange arrow initially starts high, then it decays, and then it disappears. So this does not repeat indefinitely, but the spiking activity generated by the green pulse does. It builds up precisely in the way that we saw in the laser and in the model. So after 3 or 4 spikes, it's reached its maximum amplitude, and this will just stay now for a very long time. And as you can see here, that this is actually even more stable than the laser. So I elicit some spike down here, and then it locks on pretty tightly. So this is the one spike to one delay period solution. if I change my interpulse interval, the interval between my two pulses that I apply, I can generate this 2 spike per delay period solution. So now both the spike elicited by the orange and the green arrow build up over time, and we can see this 2 spike per delay period solution here again, which looks pretty robust. I would imagine that the model predicts that over time this should give way to a solution where these spikes are separated by essentially a phase pi. This experiment didn't go on long enough to check that, but that is something to check once I get back into the lab. But essentially all of this is evidence that these solutions do exist, they coexist and are both stable. So these are both solutions for the same parameter values, the same value of kappa, the same value of tau. And I actually even tried doing a 3 spike solution, which is not shown here, but that also gave me something very, very similar. Okay, so that's essentially all I really wanted to say. So just to summarize that, so memories, and by memories, I really mean persistent spiking patterns over some delay, can be encoded in these just a single cell with a delayed AUTAPS. The system is multi-stable, so we can support different kinds of rhythms under different variation of coupling parameters, and in particular the delay period. So really, to me, this is I think of these as little memories, little packets of memories that can be stored by just a single cell. And as well as the stable branches that we see, there are also loads and loads of unstable branches that are only weakly unstable. So we can stay close to them for very long periods of time. And actually, I think there is evidence to suggest that there are other stable branches of solutions that don't have equispaced pulses. So there are probably even more stable solutions than we've looked at here. And in the future, we'll be trying to unravel a little bit more of that, exploring what happens with different underlying bifurcation structures. And more importantly, I think to the neuroscientific community, what happens when you couple multiple cells? How does the sort of scaling of this multi-stability, how does that change when I increase the number of cells? So with that, I just want to thank the people I worked with on this. So Krasi and Bernd, who provided a lot of some interesting discussions and input about how the system works, and Piotr and James, who helped out a lot with the DD-VIF tool, and Andy, who helped me build my rig in the first place so that I could do the experiment. Thanks very much.
Thank you very much, Kyle. Perfect timing. Excellent talk. Yeah, we see some comments in the chat.
I think Jonathan has a question for Kyle. I'll unmute him so he can ask him directly.
Yes.
Thanks, Daniele. Thanks, Kyle.
Hi, John.
Nice talk. Hi. So thinking in terms of memory, how do you interpret this sort of multi-stability? Do you interpret these spikes when you have more than one spike sort of per period? Do you interpret them as part of the same memory or a sort of multiplexing? Can you comment on that?
That's a great question. I guess I haven't really thought too much about multiplexing. I guess it could be. I mean, in the laser community, certainly they do play these kind of games where they start on a pattern, a rhythm, if you like, so multiple pulses per delay period, and they show that you can switch on and switch off different spikes within that. So in that regard, I guess you would think of them as multiplexing, because you could think of these extraneous perturbations you get as coming from different sort of different afferent inputs. In terms of the single cell model, I tend to think of it more as the memory is the whole spike packet itself. So you know the memory is essentially encoded in the inter-spike interval that we see in this delay period. But I haven't really explored that very much. So that's sort of my hypothesis at the minute.
Okay, thanks.
Daniele?
Yes, Kyle, very briefly. So it seems to me that the computational part used Maurice Lecar, but I wonder, you know, and of course this does a good job in reproducing what the cell does in this context. How far can you push that? Is there an even simpler model, or what makes it work of the Maurice Lecar?
That is another great question. I think the key thing is, I guess, if we think about it from a mathematical context, probably what's more important is understanding what nearby bifurcations are in the non-coupled, non-delayed system. And with that in mind, a lot of the dynamics that we're seeing, well, if I can show you, actually I can go back to the actual bifurcation diagram, which is somewhere. So I think in this particular scenario here, which I think is the closest to the experimental data that we saw, a lot of this is being organized by behavior near homoclinic bifurcations over here. So I think any kind of model that supported a sort of homoclinic bifurcation or a SNCC bifurcation, we would probably be able to see similar kind of results. I will say that we also looked at the scenario of dynamics near a hop bifurcation, and we saw similar but different results. So we could still see multi-stability. Sorry, I'm just going to a slide here, which is right at the end. We did see multi-stability. So again, here, the number here is reporting the number of stable patterns that we see in each of these regions. But now the bifurcations that switch us between different solutions are different. So there's a fold here, as we saw before, but there's some hop bifurcations, there's some torus bifurcations. So the scenario is a little bit more complicated. But I would say that any system that supports a homoclinic, for sure. Systems that with hops, I guess so. I mean, I guess probably you'd want to know about whether things like integrate and fire models can do it. I'd say probably, but I'm not completely 100% sure.
So can we move to another question, which is actually, I think, very important. On your last slide for the experimental data, can you comment on whether this is average over cells, or it's the same cell, or yeah?
So these three experiments on this slide are all in the same cell. The one on the previous slide was a different cell. So I think in total, I tried maybe 4 or 5 different cells and saw quite similar things. I mean, I think that is one thing that I think I would like to stress is that, you know, these data are not highly tuned and sort of this was the cell that I got it to work for. It was reasonably easy amongst the sort of 4 or 5 cells that I tried. Of course, the cells have a different intrinsic properties, so the results in terms of exact timings and exact parameters are slightly different. But no, this is all in one cell.
And one more related question, final one, is what would be from Victor Matveyev, what would be the metabolic limit on spike per delay per unit time?
That is another great question. The short answer is I haven't really thought about it. Obviously, there will be one. I think a great thing, I think, for us to extend the modeling, and we could extend it to include some metabolic costs of actually eliciting all of these spikes, and we can try and find a limit there. I would hope that we see the limit in kind of a smooth way and we don't just fry the cell. But it's a great question. I haven't really thought about it too much, but I think it would be great to incorporate it into the modeling into the technical cell.
With that, I'll thank again Kyle. Great talk, Victor says. And we'll move to the next speaker.
Okay, thanks very much for all the questions.
Thank you. So Jonathan, if you can share your screen now.
How's that?
Yeah, excellent. Maybe you want to put it in the presenter mode.
Yeah.
Yes, excellent. So we continue with a talk by Jonathan Rubin from the University of Pittsburgh, and he's going to talk today about the role of chloride dynamics in neuronal integration of multiple input streams. Jonathan.
Okay. Yeah, thanks, Krazi, and thanks, Daniele and the other organizers for doing all the massive sort of work to put something like this together, this new format. So the motivation for what I'm speaking about today comes from, work on the basal ganglia. Okay, so I have here just a cartoon of a coronal section through the brain, and you can see various brainstem areas that together constitute the basal ganglia. So we have the globus pallidus, for example, substantia nigra, and basal ganglia gets a lot of attention because it is a major site for the release of dopamine, a major reinforcement chemical. So it's associated with reinforcement learning and reward. And it's hypothesized to play important roles in action selection and decision making. So this is, for example, one of the subcortical structures involved in decision-making that was briefly mentioned in Ann Churchland's talk this morning. So in the picture on the right, we have these various light gray regions, which are parts of this distributed basal ganglia complex. And the idea here is that this is a framework we can think about for action selection. So if we start with inputs from the cortex, representing some sort of evidence accumulation for different decision or action possibilities, you can see there are different color-coded pathways through the basal ganglia, right? So in green, we have what's known as the direct pathway. And notice that most of the connections in this network are labeled as inhibitory, right? So once the direct pathway striatal neurons are activated, their job is to try to inhibit the downstream output from the basal ganglia. Some of you will know this as GPI. In rodent, the dominant component here is the substantia nigra pars reticulata, SNR. And so the SNR in general is inhibiting sort of motor pathways outside basal ganglia. So it's trying to prevent premature action, let's say. And so the direct pathway is trying to shut down that inhibition and allow the action to take place. Okay, but meanwhile, it's competing with the other side of the flow here. So we have this excitation coming in from STN, which is trying to keep the SNR activity strong and block actions. Okay, so meanwhile, the SNR is also integrating inhibitory inputs from the GPE. Right, so we sort of have this decision maker down at the bottom of the basal ganglia that's having to integrate all of these different input streams, and as a result, adjust its level of inhibitory output, which gates movement in this theory. Okay, great. So if we try to simulate a model of this process. Right, so over here in the schematic, it's different color coding and so on. Now CD is the striatal component, and I haven't separated out direct and indirect, so just sort of pretend they're the same population. So we see that the cortical evidence that I mentioned in the previous slide is accumulating, and that's being reflected in the ramping up activity up in the input here in the striatum. Okay, and then we have the competition that I mentioned. Right, so this striatal input ramping is sort of directly projecting to the output down here at SNR and trying to shut it off so that we can act. But meanwhile, the GPE, right, which is a source of inhibition to SNR, this is ramping down because it's also getting inhibited by the striatal input. Right, that's this pathway here. So we have a loss of inhibition on the one side, a gain of inhibition on the other side, that's this red pathway. Okay, and so those are sort of balancing, plus the loss of inhibition is complemented by a gain of excitation from the STN. So altogether, we have growing inhibition, but also decaying inhibition and growing excitation, all being integrated at the basal ganglia output, and we sort of have this balance, and we can't act until the balance is somehow broken. Okay, so in this model, the way that the balance ends up being broken is eventually the GPE firing gets so low that it can't really get much weaker anymore, whereas the direct pathway activity keeps growing. And this continued growth of inhibition eventually wins. It causes this pause in the SNR. Okay, and just ignore the magenta and cyan, please. The green and the blue represent the two different actions, so left and right. And they're competing, but you can see the evidence here was stronger for left. So eventually the blue part of the SNR, the blue action channel pauses, and we get action, right? So the downstream area, which isn't pictured here, can finally activate. Here in this case, this SC stands for superior colliculus. This is a saccade task, and so we get our action. Okay, so that's all the theory that motivates this project. But there is, as we say in the US, a wrench in the works. Okay, so there's this paper from a few years ago by Fries et al. showing what happens with some optogenetic experiments done in the basal ganglia. Okay, and this is rodent. And with optogenetics, the experimentalists could stimulate and induce firing in either the direct pathway or the indirect pathway in the basal ganglia. Okay, so remember, the direct pathway is direct. So it's, you stimulate striatum and you expect to inhibit your downstream target. Okay, and they did see that in some of their trials. So we have this collection of trials under inhibition here, all of these rasters showing this nice pause in SNR activity, and you see the mean firing rate dropping, indicated a couple of different ways. However, they also saw that with the same sort of optostimulation, some of the SNR cells responded with excitation. Okay, so here we see the firing rates are going up. Okay, completely counter to what we would think of from an inhibitory pathway. Okay, similarly, from the indirect pathway, we expect a net excitation because we're removing inhibition from GPE and we're causing firing from STN. So we expect excitation. We see that in some of the rasters, but they also recorded plenty of cells where the indirect pathway stimulation caused inhibition down at the SNR. And you can see that in the far right here. Okay, so this is at odds with the idea that these are inhibitory pathways. They are known to be GABAergic pathways, but this got us wondering what's really going on here with the integration of these different input streams, especially because notice a lot of the firing rates here, even in the absence of optogenetic stimulation, are rather high, right? So we have these sort of 15 hertz, 40 hertz, right? In fact, these areas in the basal ganglia, the GP, for example, can be firing just sort of spiking away at many tens of hertz under baseline conditions. Similarly for the SNR. So there's a lot of possible factors that could be changing the efficacy of the synapses in the midst of all of these spikes that would completely change our interpretation of the system. Okay, so the study that I'm telling you about today is brand new. I haven't given this talk before. It's currently in revision, and you can see the title here. Okay, and the results that I'm going to go through to varying extents are listed here on the right. Okay, so we think, according to our modeling, that in fact chloride dynamics are a major factor in these diverse SNR responses that are seen. Okay, we're going to look at how ongoing GPE activity at this high rate can have significant impact on various features of SNR firing, not just firing rate. So I'm going to talk about oscillations and synchrony, and then I'm going to tie this back into the action selection theory near the end. And we've done a lot of different tests of robustness. I will admit these weren't all on our initiative. Some excellent reviewers prompted a lot of these robustness studies, and things here are very robust. I just won't show most of the robustness results. Okay, so in the study, we do this, it's a computational model. It's just 2 compartments, but we do have 2 compartments, which is essential for this because the GPE inhibition actually tends to be somatic or at least proximal. So we model it in that way. And then the striatal inhibition to the SNR neuron is much more distal. Okay, so we have a 2-compartment model. Okay, and in addition to all the usual sort of Hodgkin-Huxley type equations that I'm not putting on the slide, we have chloride dynamics. We have chloride influx through the synaptic channels, these GABAergic channels. And we also have the KCC2 pump, which regulates potassium and chloride coming in and out of the cell. So the pump is trying to sort of keep pushing the chloride back out to maintain the balance inside the cell. Okay, so you can see here our chloride differential equation, the intracellular chloride concentration. It has the 2 terms that I just mentioned, the GABAergic term and the KCC2 term. We have to have a conversion factor that I couldn't fit on the slide, but the reason for that is the GABA reversal potential is actually set not just by chloride, but also by bicarbonate. Okay, and so this conversion factor makes sure that we're sort of adjusting for that and just taking into account the chloride part when we track the chloride concentration. Okay, so that's the setup. We do have, I should say, short-term synaptic depression and facilitation in the model. Okay, but these do not account for the changes that we see in the different SNR responses. Okay, that would be a natural theory, right? Think, okay, well, the synapses start to depress, that can change everything, they can facilitate, but that doesn't account for the effect. Okay, so the GITIS lab, so our collaborators at CMU, did their own optogenetic experiments, and you can see some of those summarized on the left. Okay, so we have here, it was optogenetic stimulation of the actual terminals of the GPE neurons in the vicinity of SNR. So we're making sure that they're activating the GPE terminals that release GABA to the SNR. And you can see this diversity of responses, right? So these are just examples on the top. And then the summary data is in the bar chart down below. So you can see, for example, a very small number of complete inhibition events, like the top one in the panel A1. Lots of partial inhibition, so those could be slowing down without any loss of spiking, or it could be a sustained loss of spiking, which gradually sort of makes way to some spiking. And then we also have excitation effects and a few biphasic effects, where we transition from reduced firing rate to actually increased firing rate. Okay, so this is an extremely diverse set of responses for a pathway that is just always thought of as inhibitory. You stimulate the presynaptic neuron, you have GABA release, postsynaptically you get this whole hodgepodge of things. So that's quite interesting. Now in the model, we looked at a little 2-parameter study, right? And we look at how, as we vary the strength of the pump and the GABA conductance, we can explore this space where here we have color coding by the reversal potential of GABA that results. And so we can get all the same different types of responses by sort of varying where we are in this space. So for example, if we make the pump really strong, right, and that can be enough to maintain the chloride balance, and we maintain the complete inhibition that you see in panel B here. Whoops, I think I just advanced my slide. Okay, GPE, right. Okay, on the other hand, if we reduce the pump strength somewhat, but turn up the GABA, the sort of overall chloride tone, we can balance the effects and get no effect. Or if we go even more extremely in that direction, we're actually switching the GABA reversal potential up to -50. You can see that by the red curve over here. And that's enough to switch the response to excitation. We can also get the partial inhibition types. Okay, and you can see in this partial inhibition, the time scale is long enough that we do get this accumulation of chloride, which gives us a gradual increase in eGABA. And that gradual increase makes eGABA less effective at hyperpolarizing the cell, so we get this partial inhibition. Okay, so we get similar effects, sorry about that, with striatal stimulation. This is one of those spam calls. So with striatal stimulation on the experimental side, things look similar, but you can see there are far more biphasic responses, right? So we start off with inhibition and then we get a switch over to excitation. Okay, and sure enough, we never really saw that in the model with our GPE inputs, which were coming into the soma, but when we have the inputs coming into the dendrite, and which has a different volume and so on, then we can see it's easier for the reversal potential of GABA to change significantly. And so that tends to bias us towards partial inhibition or even biphasic responses like you see here in F. Okay, so just looking at that sort of exploration of parameter space, we can at least reproduce the experimental results from various frequencies of optogenetic stimulation. Okay, so now let's go on and think about what this will actually do to the SNR dynamics. Okay, so to do this, we're going to look at PRCs. Okay, and when E-GABA is hyperpolarized. So here we have an example with E-GABA at -60. The SNR membrane potential throughout a spike, so here we have a membrane potential in blue, always sits above E-GABA, so E-GABA is having this hyperpolarizing effect. And so that will end up delaying the next action potential. If a stimulus comes in between spikes, the spike that would have occurred at this gray time is now delayed to the blue time. Okay, similarly, if we turn E-GABA up, which we now know that we can do through the chloride, the naturally occurring chloride dynamics, the same stimulus timing can lead to a spike advance, because now the E-GABA is depolarized relative to the membrane potential at that timing. Okay, so we don't have any fancy sort of nonlinearities here that cause sort of counterintuitive advance or delay. All of the switching here is due to E-GABA. So if by hand, right, we were to just push E-GABA up and down, we can look at how stimuli at different phases would encounter different relationships of E-GABA to the membrane potential, and therefore determine whether they'd be depolarizing or hyperpolarizing. And we can summarize all of that here in the lower right. Right, so we have our different advance delay sort of phase response curves based on different choices of E-GABA. Okay, now Jiang and Gutkin looked at this effect already in a paper a while ago doing weak coupling analysis. So here we are not in the weak coupling regime. We're just, for this moment, just drawing these PRCs based on the effect that a full strength stimulus would have. And here are the numerical versions of these, right? So again, I have the same sort of diagram where we have the range of possible PRCs depending on E-GABA in light gray, okay? In each individual panel, 1, 2, or 3, we've picked out a specific E-GABA, fixed that, drawn that PRC in blue, and we can see where the fixed point is predicted to be. And then we can run ongoing simulations. And here we just have a feed forward circuit, and we can see where the phases end up distributing. And they're all along the PRC, okay? And you'll see in the next slide that in fact the phases will accumulate at the fixed point when it is stable, which is what's indicated here in green. Okay, so again, when we tune E-GABA, we move the position of the fixed point, which determines the phase relationship between these two cells, okay? And we change its stability. Now in a bidirectional pair of neurons, they're both stimulating each other. The PRCs change with E-GABA, that's what's shown in blue and red here, but the fixed point location pretty much stays put, and it remains stable. So summarizing all of that, and I'm sorry, I'm realizing there's sort of a lot of information here on some of these slides, but what we've done here in each of these two very simple circuits is just plot the phase difference between the two cells in ongoing runs of the system, just letting things run and letting them kick each other with these GABAergic transmitter releases. Okay, you can see that when E-GABA is sufficiently hyperpolarized, we have a phase slipping effect. Right, so usually the phase difference between these two neurons is sort of up here close to 1 or equivalently 0. So most of the time they're close to phase locked, but every so often the phase will slip, and that's what's also shown over here. And you can see in this case, 13 successive spike pairs between the two neurons, you get this gradual transition in the timing of the two neurons relative to each other until they sync back up again. Okay, and as we depolarize E-GABA, eventually we stabilize a fixed point and we lock the two neurons, and you can see the position of the locking changes with E-GABA. With the two cell network, it's not nearly as interesting. They're just pretty much always locking at anti-phase. Okay, I'm just checking the time. Okay, so let me move on. So basically, we can vary things. This is still on the feed forward network now. So we can vary things so that the two cells have different firing rates, and we can again get phase advance or phase slip types of relationships, or phase locking, depending on how we tune E-GABA. Okay, so I'm just going to go ahead and breeze past that. Perhaps most interestingly, we then simulate a full SNR network. So 100 SNR neurons, okay, not the size of the full SNR, but a lot more than 2. Okay, we followed information on the sparse connectivity. So SNR neurons are connected to each other, which I should say is what I'm showing here. This is within SNR, and it's GPE that's setting E-GABA. Okay, so I'm no longer looking at GPE's inhibition of SNR. Okay, so within the network, we have this GPE, which is going to set E-GABA. And then we have connections between the SNR neurons, which are fairly sparse. And again, as we tune E-GABA, we can see radical changes in the variability of SNR firing and what these rasters look like. And we can plot spectrograms over low frequency ranges and see that E-GABA drastically tunes the oscillatory power in the network. So we can be in a sort of phase slipping mode or a phase advancing mode at the 2 extremes. And in between, we don't have oscillations. Okay, and so this is exciting because oscillations in the basal ganglia are associated with a lot of different disorders. This is not the range of frequencies that people usually think of. Right? Beta is sort of the superstar frequency range right now, which is more like 15 to 30 or maybe down to 8 to 30 hertz. This is 0 to 4, but we have a brand new paper also from the Gittes lab showing that in these animals, where all this data is from, in the mouse, there isn't really beta. Other people have shown that as well. And we do find significant low frequency oscillations in the SNR under dopamine depletion. Okay, and that's what this data shows, a couple of examples of what the power spectra look like. And here the total fraction of units with various forms of dopamine depletion showing the low frequency oscillations, and none of them are showing beta. Okay, and these persist with M1 lesions. So these are really coming from the basal ganglia. Okay, so basically what I've shown you is tuning the ongoing activity from GPE could adjust E-GABA in such a way that it changes the ability, whether SNR ends up producing oscillations or not. Okay, there's a prediction associated with that, which is if we stimulate GPE under dopamine depletion, we should be able to turn these oscillations off. And we did that, okay, but I'm out of time, so I'm just going to show you the answer here is yes, okay, that prediction works. And finally, coming back to the action selection, okay, the idea in the action selection scenario, remember, is that this lower frequency striatal activity is working to pause the SNR, right? It has to be this inhibitory input that can pause the SNR and allow us to act. And now if we have the GPE sort of setting the E-GABA, we can change the effectiveness of this inhibition. So we can change its ability to pause the SNR. So we could delay our action selection by tuning the GPE, right? So as we vary the sort of chloride load in the soma or the dendrite, we change E-GABA. Here's 2 examples here where we see with different thresholds needed to initiate action, we get different timings of crossings. And you can see the results on the lower right, although I'm flying now. The time to act goes up with the overall sort of GABA conductance and chloride load. Okay, so the summary is here. We find in our computational modeling that changes in chloride load can alter these GABA effects on functional timescales. We think that this could be a mechanism, so a sort of different interpretation of what the indirect pathway is doing, where it could tune synchrony oscillations and a sort of threshold for decision making. And as I mentioned earlier, the effects are robust to a whole lot of different things that I didn't talk about today. So thanks for your attention. Sorry to accelerate throughout the talk. But
thank you very much, John. Really, really interesting. And very, very clear talk. Thank you. Are there any questions? If not, I can ask you a question because it is very thought-provoking, actually. This pause in the action, the timing to action, and this indirect and direct pathways, do you think this might be involved in freezing of gait in Parkinson's?
Yeah, so yes, I think that's a great point, and I'm tentatively, like hesitantly, but in agreement, right? I don't want to jump to any big conclusions, but it really makes sense, right? If we need, at least for certain movements, this pausing mechanism, and dopamine depletion has caused GPE firing rate to change, it could be changing the chloride load, which could make it much harder to do this pause.
Yeah.
So, yeah, spot on.
Yeah, because there has been this hypothesis that this indirect pathway is involved in freezing of gates specifically in Parkinson's, but nobody has come up with any mechanism. So when I was listening to your talk, it was really kind of, so very, very interesting. Somebody, Taylor Cow says, looking forward to the paper. So hopefully it's going to come soon. I will definitely be reading it. Any other questions?
I hope that as well.
Yeah. So we have a question here. Would you expect the results to change if you could scale your model to full size?
Okay, so first let me say no, because 100 neurons was a lot more than 2, and we did have sort of the right degree of connections within that model. So I think at least qualitatively, I'd like to believe, that we'd see something similar. Since the door is open, I'll mention we used sort of the mouse firing rates, which are lower than primate. But one of the things we did test for heterogeneity is if we jacked up the firing rates. So now we have 30 hertz instead of 10 hertz at baseline. We do still get oscillations. The frequencies can sort of switch and become a little higher. So, yeah.
Yeah. We have another question from Victor. Do you think there could be a simple pharmacological way to verify the role of chloride channels on the dynamics of I think it's GABA regulation? Yeah.
Yeah. So simple, maybe not, but reasonable, yes. And so now this is on the to-do list for the Gittes lab, but with the shutdown and everything, the timetable for that is probably not very rapid.
One more question by Dmitry Todorov, that last one. In general, do you think in actual disease, this mechanism is robust enough to account for oscillations despite high variability in patients?
Yeah, so that's, of course, a key question. I think this is probably a factor. I'm not going to say this is the source of oscillations, and I'm not even going to say, like there's one frequency band, but people do see delta oscillations in humans, and they've been overlooked until now in favor of beta. But this whole collection of work says to us, hey, maybe a little more attention should be paid to delta oscillations as well.
Okay, so thank you very much. We have to move on to the next speaker. I'd like to invite James. Oh, hello, James.
Hey, Grazi. Good morning, everybody else.
Good morning.
Thanks for your talk, Jonathan. I'm sorry I missed everybody else's talks, but I was snoring.
So, James, it would be great if you could share your screen.
Actually, David's going to be the one who's going to be sharing his screen.
Oh, hello, David.
Hello, Grazi. How are you? I'll share my screen.
Yes, thank you. Yeah, so actually we're having a double act. I'm going to introduce in a minute when the... Okay, excellent.
We good?
Yeah, so our final talk for the session is a double act, as I mentioned, from New Zealand.
Sorry, one
US.
second. David, we can see both your current slide and your future slide.
That's unfortunate.
Now it's great.
I think you might want to try and
Turn off the presenter view, maybe.
yes. Crazy to have interrupted you.
No, I can repeat it.
Can you see?
So actually we do see the next slide as well for some reason.
Can you try and press Control X or Apple X after you share a screen?
Okay, let's try that. You're still seeing both screens?
Yeah.
Let me stop sharing this and do this.
This is usually when you're connected to a projector or something, isn't it?
No, I have two monitors, but
Probably that's why.
still?
Yeah.
Yeah. But if you're not in presenter mode, actually, it's
Yeah, we can do it like that.
You know, do it like this.
As big as possible. Yeah, that is pretty. That's almost the same as otherwise.
That's
You know,
pretty.
Slideshow. Yeah, it's not in presenter mode, but we go with this.
Yeah. It's
If that's
just
okay, if that's okay, David, unless you'd rather I shared screen and ran the slides.
I mean, you can do that as well, actually. I mean, I'll just say next slide, play the movie, whatever. Okay, you want to try that? I'll exit this.
Okay. Let's see if this works.
Victor is suggesting. Let's see whether this works.
How's that working?
Yes,
Perfect.
great.
It's good, is it?
Yes, perfect.
Leave it to a mathematician.
Absolutely. We are such practical people.
Yeah.
So I'll start again. It's really my pleasure to welcome James and David. David, I haven't seen since my PhD, which is really exciting, actually. That was a great time when I visited Rochester. So yeah, we have this double act from between New Zealand and the US, which is amazing. And this could be done virtually. So I'm really looking forward to hearing your talk, David and James, on calcium dynamics in salivary glands, modeling and experiments.
Great. Thanks very much, Krazi. It's wonderful to be here, even at such an awful hour of the morning, which it is for me. David's going to be talking about our experimental data. When I say our experimental data, of course, I really mean his experimental data. And I'll be talking about the models. So since you always have to start with the data before you do a model, David is going to take over the first part of the talk. And so, David, over to you.
Okay. So all the data that we're going to present is actually from exocrine glands. And so what the models have been trying to interrogate is stimulus secretion coupling in exocrine glands. So these glands are, you can think of them like a bunch of grapes, essentially, where one cell type, the acinus, is the grape, and the other cell type, the ducts, is the stalk. And the cells take they take stimulus onto the acinus, and they secrete from the acinus into a ductal system, which ends up secreting fluid and enzymes into your mouth or into your gut. So next slide. Over the last 25 years or so, James and I's collaboration has been to model fluid secretion in the salivary gland. And essentially, without going over all the details, what the gland is trying to do is move chloride from the blood into the ductal system. And that will allow sodium to follow and water to follow the sodium chloride, giving you a saliva which is sodium chloride rich. So next slide. The impetus for this secretion is obviously the sight, the smell, and the thought of food. So when those things occur, next slide, there is a nervous input through the parasympathetic nervous system, which releases acetylcholine, next slide, and that causes an increase in calcium within the acinus cell, which actually activates that chloride channel and gets the whole system working. So next slide. So we have gathered a bunch of information that James has used in the modeling over the years. And this has been in the form of gathering spatial information, through calcium imaging experiments and temporal information by looking at the calcium signal actually in cells. And the principles that we've established that the model has been built on is that there's both spatial and temporal information. And the fundamental facts are that we get long, slow calcium oscillations when we stimulate with the neurotransmitter. So in the right panel, you see cells being stimulated with carbachol, which is an acetylcholine analog. And we essentially get long, slow calcium oscillations. And the other fundamental characteristic we can see from the movies are that we get global calcium signals. So the signals have spatial information. So next slide. To illustrate these global calcium signals, that these signals are invariably global, what we actually do is we produce the stimulus for the calcium signal in one end of the cell. And so that's essentially releasing IP3 using a laser in a small volume in the blood face of the cell. And if James plays the movie again, you'll basically see that even though we produced the calcium signal, we initiated the signal in the basolateral phase, the majority of the signal initiates in the apical phase, but invariably becomes global. So these factors were actually the fundamental basis of the model that James has developed over the last 5 or so years. Next slide.
All right, so that's where I come in. That's the basic experimental information. And the question is, how do we write down a model of all this stuff? Actually, the first question one should always ask, of course, is why should we model it? But our modeling goal is to understand what on earth is going on. So let me give you a very brief primer on how to model calcium in cells. I'm sure most of you know this already, but for those who don't, here's a very brief introduction. The cytoplasm of a cell has a very low calcium concentration, extremely low. That's because calcium is in fact, or at least free calcium ions are in fact toxic. They turn stuff on and they turn stuff off. And if the calcium goes up too high for too long, the cell will die. And so the cell spends a lot of energy keeping its cytoplasmic calcium concentration very low. On the other hand, the calcium outside the cell is extremely high. This is about 4 or 5 orders of magnitude bigger than the calcium in the cytoplasm. Not to mention that the endoplasmic reticulum also has a very high calcium concentration, as does, well, the mitochondria is not all that high, to be honest. So that means that the cytoplasm is under intense calcium pressure. And all you have to do is open up these calcium channels, open up a leak here, say, or open up an IP3 channel, IP3 receptor, which is a calcium channel, or a ryanodine receptor, which is also a calcium channel. You open up those channels just a little bit and calcium rushes in, which means that the cytoplasmic concentration goes right the way up. Very fast. Well, you know, not fast compared to neurons, for example, but fast compared to, I don't know, the speed at which I run, for example. Then in order to get that calcium concentration back down, you just have to shut these channels. So these channels go shut, and then circuit pumps, sarcoplasmic and endoplasmic reticular calcium ATPases, take the calcium out of the cytoplasm and put it back into the ER, or they take it outside of the cell. And by this sequential opening of calcium channels and then reuptake by the calcium ATPase pumps, you get the calcium concentration in the cell going up and then down and up and then down. And it is dogma that the signal is carried by the frequency of that oscillation of calcium. So, you know, a fast one is sends the regulation network a slightly different signal than does a slow one. But we shall talk more about that in a minute. I say it's dogma, but does not actually appear to be true for our cells here. You construct the model, you take each of these various bits and pieces, you know, the pumps and the calcium fluxes in and the ryanodine receptor and the IP3 receptor, and each of those has its own somewhat complicated model, which people have worked on over the years, and you decide which bits to put in, and you grab those models and you stick them all together, you connect them up, and that gives you your overall model. So for example, for our cells, for parotid acinar cells, we know that you have to have circuit pumps and plasma membrane pumps. You've got to have store operated calcium channels, and you've got to have an IP3 receptor. You also have to have buffers, but I didn't put that arrow in. I'm not sure why I didn't, but I didn't. And most of these other things are probably not as important for the kinds of questions we're asking here. So you build the model in a modular kind of approach, so it can be adapted to many different other cells as and when needed. That's our calcium toolbox. So how do these cells really work? I talked about the calcium so far, but there's all the other ions, the chloride, the sodium, and the potassium ions, not to mention the bicarbonate and protons, which are really, really important. As David said, it is the transport of chloride ions from the outside of the cell, from the interstitium into the cell, and thence into the lumen. It's that transport of chloride followed by the flux of sodium that concentrates sodium chloride in the lumen, and that draws water from the interstitium through the cell into the lumen by osmosis. And therefore cells have got to have very well regulated ion transporters to make sure they keep their volume stable and to make sure that they can transport chloride as and when required. So in the basal membrane of the cell, which is the one facing the interstitium, you have a sodium-potassium chloride transporter exchanger, you have a sodium-proton exchanger, you have an anion exchanger, all these bits and pieces. You have sodium-potassium ATPase, you have a calcium-activated potassium channel, etc., etc. And in the basal membrane of the cell, you have calcium-activated chloride channels. So you have rather different sets of channels in the apical membrane than you do in the basal membrane. And of course, you have bicarbonate buffering and pH control in the middle of the cell. For the calcium, you have the IP3 receptors sitting close to the apical membrane of the cell. We know that's where they sit because David did a wonderful experiment where he labeled them with his high performance, super duper, extra special plus highly expensive microscope, and all the IP3 receptors. You could see the little green blobs sitting really, really, really close to the little red blobs, which were these chloride channels. So most of what I say is in fact well justified by detailed experimental evidence, although I'm quite sure not all of it is justified by evidence, as is my way. Okay, and so when you get calcium released from the IP3 receptors, which are sitting in this part of the cell, that creates the back propagation of a calcium wave back through the cell in order to stimulate PLC, phospholipase C, which makes IP3 in the basal membrane of the cell. Therefore, according to our thinking, the back propagation of this calcium wave is vital for how these cells secrete water. So here's we'll just briefly go through that. Here's the basal region. Here's the apical region. When you bind agonist, it makes IP3 in the basal region. That makes IP3, diffuses through the cell. It then binds the IP3 receptors in the apical region of the cell. Those IP3 receptors release calcium. So calcium is released in the apical region. That stimulates a back-propagated calcium wave, which goes back to the basal end of the cell, and that lets us stimulate PLC and make an oscillation of calcium. So that was our current thinking, and in fact, that's sort of the dogma in the field, and it's been that way for many years. What else did we do? Well, we actually built this into a full three-dimensional reconstruction. Again, David used a super duper fancy, expensive microscope, which has a special name, which I can't remember, and took optical slices through a group of 7 cells. And we reconstructed those optical slices into a three-dimensional finite element reconstruction of those 7 cells, which is this group here, and together with the associated lumen. So this is the lumen that collects the water. These are the cells which are clustered around this bit of lumen. Here's the original data on the left. And what you can see is that each cell is sort of surrounded by the lumen. There are fingers of it. It's like the cell is a fist and the lumen is a bunch of fingers which sort of go around it like this. And this is the case for every cell. And each part of this lumen is shared between, in general, 2 cells. We then solved our model equations on that group of cells, and we tried to understand how our mechanism was working to secrete secrete saliva. These are typical results. We could show a lot more, of course. Here's a lovely movie by John Rugas, who has done lots of the programming for what you see here. I didn't write the programs, of course. I get paid far too much to write programs, so we hire someone else to write programs. Many years ago, it was Krasi's turn to write programs for me. Thank you, Krasi. But now it's somebody else's. Here are typical responses from the calcium oscillations from the apical region, from the basal region, shown here. Here's the rate of fluid flow from the cluster. As you can see, you get this weird sort of MMO here. That's only because you have 7 cells, each of which is oscillating at a slightly different frequency, and so you get this summation of the fluid coming out of each cell, giving you this mixed mode pattern here. And here are the concentrations in the cell and the lumen of carbon dioxide, chloride, bicarbonate, potassium, etc. So all these concentrations check out very well. The calcium oscillations check out very well. The fluid flow checks out very well. And we concluded that, well, we kind of understand what's going on, even in the 3-dimensional thing. I mean, there are questions to do with the spatial distribution of the calcium. There are still questions, but we figured we kind of understood it. That was all very well, but then came Jonathan Rubin's spanner in the works, where I head back over to David.
So, you know, the dirty little secret about all the data that we produced over the last 20 years or so, up until this point, is that it's been produced in isolated cells that we take from a gland and we digest by enzymes, and therefore it's not in its native environment. I mean, that's for sure. I mean, it has no interaction with extracellular matrix or basement membrane. And another issue with our previous data is how the agonist is actually presented to the cells. We essentially just add a square wave pulse of agonist and we monitor the response. And that's clearly not what is happening when the gland is stimulated by a nerve. So we decided that we would try and measure some of these parameters in vivo using intravital microscopy. So essentially we take an animal which is expressing a genetically encoded calcium indicator, and we use multiphoton microscopy to interrogate the fluorescence from this dye in a living mouse. So next slide.
I've forgotten I was doing the slides.
Yeah, you can play the movie as well, James. So essentially we breed a mouse that will drive the expression of the dye of the probe in one particular cell type. So this is in the acinar cells. So GCAMP6F is the indicator, the MIST is the promoter which drives this expression in acinar cells. So you can see these big black holes in the middle here. These are essentially where the ducts would be. So all we're measuring from is acinar cells. So next slide. So we can monitor the calcium signal in slabs of tissue in vivo, and we can stimulate a nerve bundle that goes into the gland with different frequencies of stimulation. Here we're stimulating with 5 Hertz stimulation to the nerve and monitoring the calcium signal in regions of interest that you can see that James is pointing to. And essentially what we see here is, again, we're seeing calcium oscillations. We're recording at 10 frames a second. And the first difference that we see is that we still get calcium oscillations, but they're actually much faster than we were seeing in isolated cells. So next slide. The next thing that is different from experiments that we did previously is if we zoom in a little bit now and stimulate with a lower frequency, now in contrast to the isolated cell experiment where we invariably saw global responses, here we're seeing highly localized responses around the apical end of the sun. You can see this with these flashes of yellow and red around the lumen. So we're getting apically limited responses, again, which are occurring quite rapidly. Again, this is recording 10 frames a second. So next slide. So we can visualize where the response is occurring by taking that whole movie and generating a standard deviation plot of the whole movie sequence of the 600 frames. And you can see if we zoom in on one particular acinus, which is probably about 10 cells, and you look at this volcano plot, again, it's pretty obvious that the signal is in the apical end of the cell and is really not globalizing to the basolateral regions of the cell. So next slide. If we measure the calcium signal again in that acinus cell and essentially just look at the fluorescence within regions which represent the apical or the basal region, you can see that you get these much faster calcium oscillations which are restricted to the apical region of the cell. They're not globalized. There's pretty much nothing going on in the basal regions of the cell. So that's another difference from the in vitro data. So next slide. We also noticed that there was heterogeneity across the field that we were looking at. So if we stimulated the glands at 3 Hertz, we'd have a portion of the cells that increased their calcium. If we increased the frequency of stimulation, we would recruit more of the field of view to respond, but the initial responding regions appear to always respond first. And so to analyze this, we decided we'd take an approach to divide the field of view into a grid. And you can see that here. We divided it into an 8x8 grid, 32 microns by 32 microns, and then essentially process the image as a standard deviation image within each of the 32 grids. So next slide. So what we did here to look at the heterogeneity across the responding grids was to essentially rank the grids based on the magnitude of the response in a particular grid. So the grid that responds greatest here is G6. It has the lightest gray, and this is analyzed from a frequency where all the cells respond. So next slide. If we now plot the same calcium response based on that same ranking for low frequency and high frequency, you can see that generally an area of the field of view responds first and biggest to all frequencies. So we're interested in why that happens. So next slide. And we rationalize that this could be that there's actually a heterogeneity of neural distribution within the field of view. That's one thing that could explain this. And so a particular area of the field of view is preferentially stimulated. The other explanation could be that there's a homogeneous nerve distribution, but as we increase the frequency of stimulation, we recruit more of those nerves to actually fire, more of those neurons to actually fire. Another alternative, I guess, is that the acinus cells could have a heterogeneous distribution of neurotransmitter receptors on the cells, such that the ones with the most receptors respond first. So we're interested in sorting this out. I'm going to tell you we don't have the answer, but next slide. To sort of at least address the first issue, that there's a heterogeneity of neural inputs, we did was actually cross a mouse that expresses membrane-bound red fluorescent protein in all cells with a driver that would put a green fluorescent protein in cells that express acetylcholine transferase, i.e. they're representative of parasympathetic neurons. And you can see that there's a relatively homogeneous distribution of neurons through the field of view. So we don't think it's that there's a heterogeneous distribution of neurons. And to get at some of the other issues, next slide, we've crossed that same, so Jane.
Was that the right one?
That's the right one. I just was worried that you hadn't had enough tea this morning. So we've crossed this GCAMP mouse with the cholinesterase transferase mouse. So now we're actually monitoring the calcium signals in the parasympathetic neurons, and we can monitor those signals. And our first indication is that that at higher frequencies, we get more cells to respond, but this is a work in progress. So presented with that new data, it's over to James to sort this out as far as the modeling goes.
Right, so there's been a lot of developments just in the last, I don't know, 2 or 3 months or something. It's very recent. None of this is published yet, I don't think, is it, David? We haven't done anything yet? No. And the problem is that David's new data have shown us quite clearly that our previous model simply doesn't work. It's just wrong. Our previous model, you might or might not remember, is that you had a slow calcium oscillation and you had to have a back propagation of a calcium wave. Without the back propagation of a calcium wave, it doesn't work. The cells don't secrete saliva. And yet when you look at it in real animals, there is no back propagated calcium wave. There is an isolated cells, but not in real animals. And therefore we actually have to construct a completely new model, which goes against the current dogma of how these cells work. And we haven't done this yet. So we actually have no answer. I mean, but this is what we think is going to happen. There is, I should add parenthetically, quite a lot of data that most of the channels and things and transporters that are in the basal membrane also sit in the apical membrane, possibly at lower density, but they seem to be there. So for example, we've shown that there's an ATPase in the apical membrane, which was unexpected. We've shown there's a calcium activated potassium channel in the apical membrane, which again was unexpected. But that means that all our previous data over the past 5 years, which was previously somewhat of a puzzle, we didn't understand why those things were in the apical membrane. We now believe we do understand why they are. And it is because the apical membrane of the cell seems to be the only part of the cell which sees a rise in calcium concentration. And therefore, anything to do with fluid secretion, all those channels, they have to sit in the apical membrane of the cell, or the cell doesn't work. And that's going to be our new hypothesis, is that in fact a parotid acinar cell, although it's a polarized cell, actually does all the work, or most of the work anyway, in the apical region, which contains all the necessary transporters and calcium bits. Will it work? We don't know. Will it transport water? We don't know. Will it be able to maintain a stable cell volume, which is quite difficult to get in a model? Again, we don't know. But I suppose time will tell. So David and I will keep our fingers crossed while we go and construct this new paradigm and submit it for publication, I would guess. And I think that is probably time, in which case we are done. Just a list of collaborators. I just do want to give a special shout out to John Rugas, who is a scientific programmer in Auckland and did a lot of the pictures that you saw today. And I think that's it from us.
Thank you very much, James. Really interesting. Maybe I can start with asking a question about your previous picture, the new view of the cell. So you have here calcium sensitive potassium channels. Why are they there?
Okay, so the reason you have a calcium activated potassium channel is because you have a calcium activated chloride channel and you want to transport chloride. So when the calcium goes up, it activates a chloride channel, which lets chloride flow through, but that would depolarize the membrane. And so if you were to not compensate for that, you would have the membrane depolarize, and that would stop calcium flux, which you don't want. So you have to re-hyperpolarize the membrane, or keep it hyperpolarized, in order to maintain calcium flow. And that's what this potassium channel does. When calcium activates the potassium channel, it then pulls the membrane potential back down and keeps the membrane potential high and negative, allowing for continued chloride flow. The dogma says that all those channels, all those calcium activated potassium channels, sit in the basal membrane of the cell. That doesn't work. The model shows quite clearly you cannot have that and have it consistent with the data from real mice. Therefore, those channels have to sit in the apical membrane of the cell, we have in fact shown experimentally that they are there previously.
But you will then have you have a response actually in the membrane potential when the cell is going to be kind of excitable. I mean,
yeah. Well, it's not actually an excitable cell, but your apical membrane potential and your basal membrane potential, they are both different, and they're both going up and down, up and down, up and down, up and down. Yeah, for sure.
Yeah, we have a question from Victor, but just before that, I want to ask one question. Sorry, Victor, to David. David, when you stimulate with this 3 hertz, 2 hertz, 1 hertz, 5 hertz, is this optogenetic stimulation?
No. Essentially, there's a nerve bundle that goes into the gland, and it's just wrapped with a platinum electrode.
So you can access it from the outside.
Yeah, we can access it from the outside. Yeah. No, optogenetic stimulation would be very elegant, and that's an interesting idea.
Actually, I would like to jump in and point out that David tried to do an experiment where he just made the mouse smell cheese and tried to get the oscillation.
I thought that would be so cool that if it smelt and salivated and we could see the calcium signals, that would be brilliant. And we tried cheese, that didn't work. We tried Japanese unami bomb, that didn't work. And so then we rationalized the fact that mice actually, that we actually didn't make them smell it. We tried to get them to taste it. And so I think that's the issue. Maybe mice don't taste, they actually do smell. So we haven't done the smell experiment, and that's on the plate.
Yes, Victor says peanut butter works best. But seriously, the question, he's asking how compartmentalized is the apical and distal parts. So how, I can't pronounce it very well, but how separate.
Compartmentalized,
yeah. Is there very special? Is there some very special cytoplasmic organization?
In our model, no, Victor, there isn't. It's a really good question. The compartmentalization is not built in directly. It's simply a result of the low diffusion coefficient of calcium. So you have a single domain where you have calcium diffusing with a very low diffusion coefficient, around about 5 to 10 microns per second. But the other ions, the chloride, potassium, and sodium ions, they all have a much higher diffusion coefficient. So effectively, the chloride, sodium, and potassium are homogeneous in the cell because they diffuse so fast. IP3 is, as we discovered, effectively homogeneous in the cell because, again, IP3 diffuses fast enough for that. But the calcium is not homogeneous in the cell because of its low diffusion coefficient. And that's all you need to have the difference between the apical and the basal regions. You, of course, have to have a resistance between the apical membrane potential and the basal membrane potential, and that resistance is provided by the tight junctions. So that's how the model is constructed.
Yeah, so morphologically, they are two distinct domains. So the tight junctions will separate the apical transporters from the basolateral transporters. They can't mix.
Right. Makes sense. I think we have a question from Kyle. Kyle, would you like to unmute yourself?
I think I am unmuted.
Oh, okay.
Here you are.
Brilliant. Thanks very much for a great talk to both James and David. I have a very rudimentary question. So in this new model on this slide, it seems like sort of most of the heavy lifting, it seems, well, at least with respect to calcium, is being done at the apical end. So what does the basal end need to actually do? I mean, does it need to be this complicated?
Well, you have to have a basal region, otherwise you can't get in chloride from the outside. After all, the cell does have to transport chloride from the interstitium into the lumen. So you've got to have a basal region for that. What else does it do? The basal region contains the receptors that bind the neurotransmitters that start the whole process off. The basal region contains the proteins that actually make IP3 that release calcium. The basal membrane contains a whole pile of other chloride reuptake mechanisms, such as the sodium chloride potassium co-transporter. And of course, the basal region contains all the bits and pieces which control pH, NHEs, these here, bicarbonate exchanges, etc. So you can't do without a basal region. You just don't need a calcium response in the basal region in order for the cell to secrete water.
And actually, it might actually safeguard the cell not having a calcium signal in a sustained calcium signal in the basolateral region. The nucleus is jammed up against the basolateral membrane, and the sustained calcium signal in the nucleus is probably not a good thing.
Yeah, absolutely.
Okay, well, I guess if there are no other questions, I'd like to thank all the speakers in the session. Really enjoyed all the talks, very interesting. Thank you very much, and especially for James for getting up so early. It was really nice to see you. And David, you too as well. It's incredible. So thank you all, and I hope you enjoyed it for the participants. I think Daniela would like to say a few words.
I also want to thank all the speakers of today's session, and I just want to remind everybody that now we are at a crucial point at ICMNS. There are posters that started 8 minutes ago. So what I would urge you to do is to take your booklets, pick up a selection of posters and click on the Zoom links. And and let's show some enthusiasm for the poster session as well. Thank you very much. I'm now closing the YouTube session, and see you tomorrow morning. Goodbye.
Thank you. Thank you all.